jumping spider Paraphidippus aurantius, Araneae Salticidae, of the Ecological Reserve of Pedregal de San Ángel, Ciudad de Mexico.
Yes, I'm here. Can you hear me well?
Yes, we can hear you. You can share.
Okay. Can you see my screen?
Yeah.
Okay, perfect. Thanks for that introduction. Okay, I'm gonna present a contribution to the life history of jumping spider Paraphidipusarantis of the Ecological Reserve of Pedregal de San Ángel, Ciudad de Mexico. The study of phenology, it's important to understand how the environment, including biotic and abiotic conditions, affect the development and the life cycle of species. Between jumping spiders exist considerable differences about the preference of niches and the tolerance of physiological boundaries where the species could live. For example, in temperate and tropical climates, the life cycles could be annual or biennial, while the breeding season could be in spring, summer, or autumn. At the present time, the studies of life histories of jumping spiders are very limited to works done in 40s, 80s, and in the beginnings of 2000s. Our target organism of study was the emerald jumping spider Paraphidipusarantis. This species has a widespread distribution from the United States to Costa Rica and the Great Antilles. In Mexico, the records include 25 of the 32 states. This jumping spider is very common and abundant in a great range of habitats, including urban areas. We can find her foraging in riparian trees, shrubs, trees, herbs, and grasses. This species is considered a medium-length spider, and the male is shiny dark, colored with iridescent green scales in the dorsum, while the females present polymorphism in the color pattern with the body totally dark or covered with metallic green scales and yellow hairs. Currently, we know a little about the biology of Paraphidipusarantis. For example, some aspects of the courtship, the fidelity of the spider to the nest in natural condition, and its response to the predation threats. However, we don't know anything about its phenology or more detailed about its life cycle. For that reason, in this study, we want to respond to the following questions. First, how is the phenology of Paraphidipusarantis in natural conditions? Second, how many polymorphism in column pattern females of a single population of Paraphidipusarantis have? And finally, does this species have parental care behavior? So, to respond to that questions, we studied for a year and a half the population of the Ecological Reserve of Pedregal de San Ángel, or REPSA, located in Mexico City. In REPSA, the soil is scarce, shallow, and with an abundant outcrop of volcanic rocks. The solar incidence and the evaporation are very high. According to Köppen's classifications, weather is temperate subhumid with a rainy season and summer. The vegetation corresponds to a hierarchy of scrub with herbs and shrubs. Also, REPSA has a very conspicuous seasonality with a rainy season between June to October and a dry season from November to May. From April 2018 to November 2019, the reserve was visited once per month by 3 collectors from 10 to 12 o'clock. At the end of the study, a total of 114 searching hours were summarized. The collecting methods include beating and active searching. A monthly census of the number of males, females, juveniles, subadults, and exact was taken. All these spiders were maintained in captivity and rearing to adulthood until its death. Additionally, the mean temperature, mean humidity percentage, and mean precipitation for each month were obtained from the ROA UNAM Weather Service. Then, the impact of the meteorological factors and seasons over the abundance and the presence of different instars of the population were analyzed. So, what we obtained? In abundance, we collected a total of 304 spiders. In general, the population has an exponential growth related with increase of temperature. As you can see in the graphics, at the beginning of the dry season, that is in November to February, the abundance was low while the temperature decreased here. In February, when the temperature increased at least 3 degrees, the abundance increased equally. At the end of the dry season and the half of the rainy season, that is in May to August, the abundance of remaining in a high and stable pattern, just here. Then when the precipitation and temperature were low again, that occurs in October, the abundance had a conspicuous decline. However, the T student test doesn't show significant difference in abundance between seasons. This abundance population pattern is like other patterns found in the insects in Repsa, like grasshoppers, flies, and collembola. So, we can say that increasing the abundance pattern of arthropods is related to increase of the vegetal coverture and the elevation of the primary productivity in the rainy season. Now, about phenology. The chi test shows that developmental stages are depends of the season. Data shows Paraphidipusarantis is a univoltine species and its life cycle is completed in a year. And it's a Ostenochronous species that reach maturity at summer during the warmer and most humid months. These life cycle strategies are common for the 45% of the spiders in temperate regions. Also, data shows that Paraphidipusarantis survives the driest and coldest months of the year as a post-embryo or early juvenile stages. These instars are recognizable because the spiders are less than 3 millimeters in length. Other jumping spiders like Lesomanes viridis or Pellegrina galatea shows survive the winter condition as post-embryos do. On the other hand, the presence of adults and subadults are related with a major availability of food resources in the rainy season. Males actually were present only in these seasons, while the females were captured in both seasons. Males reach adulthood 1 month earlier than females. This early maturity and great agility of males are life strategies that emphasize the courtship, mating, and searching of females. In this study, we don't report mating. Nevertheless, the data suggested that the breeding season was during the rainy season and listed at least 3 months when both sexes were found in the samples, from July to August, more or less. After breeding season, the males of Paraphidipusarantis died, and the abundance pattern have a fall at the end of the rainy season. Actually, the presence of males was correlated with the precipitation, and we didn't find any males in the dry season. On the other hand, at the beginning of the dry season, from September to December, we found females associated with the X-sex. Females have a longer lifetime, maybe favored by the successive oviposition phenomenon. Now, about paternal care. All females with X-sex build a nest of dense silk. Most of the time, they stayed resting over the exact capsule. And even when the food resources were available, their feeding rate was reduced to 1 point per week. This behavior is on par with the parental care conduct of other jumping spiders, where the female stayed closer to the offspring and it built a nest with at least 5 times more silk than the regular nests. This protecting behavior is useful against potential predators, parasites, and against desiccations too. Finally, what we found about polymorphism? Well, all the females presented a single color pattern, while females show 4 patterns: green with orange spots, green with white spots, black with orange spots, and black with white spots. Also, we distinguish 2 moments across the developmental growth of juvenile spiders in captivity where the color pattern changed drastically. First, when the spiderling reached more than 3 millimeters in total length, the body was covered with yellow golden hairs. And in the dorsum appeared a medial band of green metallic scales, this one. Second, when spiders move to pre-penultimate or penultimate maturity stage, individuals have dorsum totally metallic, green, or dark. And depending if the spider was a male or melanocytic female, integument was dark. In the other case, in lighter green females, integument was pale. With this evidence, so we can associate the color pattern with this seasonality. As you can see, during the dry station here in Repsa prevail the yellow, light, and brown colors of leaf litter and grasses. And it is possible seeing that the dark of volcanic rocks in the soil. So this palette of colors is the same as the genuine of Paraphidipusarantis, and the spider were dark or metallic green. So we can say allows spiders to
10 minutes.
thermoregulation in arthropods, and about the reason in hummingbirds of Maratus jumping spider. In this study, it is very difficult to say if the metallic green colors have a role in sexual selection or is just a trait linked with camouflage. So other works must explore this possibility. As a conclusion, well, the population of Paraphidipusarantis from Repsa maintained a stable pattern of abundance despite the change in resource disposition and remarkable variation in abiotic conditions between seasons. Further work should analyze the relationship between phenology of these species against the phenology of plants, crickets, grasshoppers, birds, and other predators to elucidate the role of a spider in the trophic chains in the reserve. And finally, this is the first study that documents the presence of all polymorphism known for females in a single population. Other efforts must be made to understand the genetic mechanism behind chromatic variation and the function of iridescent as adaptive character. So, thank for all of you to be in the chat, and I want to thank to the Postgrado en Ciencias Biológicas of the UNAM and CONACYT for their financial support, and the REPSA managers for the access and guard and in the study area. And I want to thank to all members of the Phenology team and the team of Viernes de Arañas.
Okay, thank you, Dariana. Great talk. You have a few questions. From Catherine Scott, do the males ward subadult females?
Can you please repeat me that?
Si los machos cuidan a las hembras subadultas.
Yeah, we can't see this behavior, but in 2017, through law, photograph this behavior. Actually, the males just guard the subadult females, and in some cases, when two males are in contact, they start like a fight for this female nest.
Another question from Dinesh Rao. There were differences in female more frequencies in the same season?
No, we don't see any difference between this morphological or this polymorphism. Actually, the next step of the study, we would like to prove if exists a difference in a fitness between the green color patterns against the dark color patterns. But in this study, at least, we don't see any difference in the frequency.
Okay, thank you. There are a lot of compliments in the chat. And there is another question. Nice work. Apparently there are no more questions.